Right. Hello, everyone. The time has just gone 3 p.m. CAT on my end, and so we will officially kick off today's session. For the benefit of those who were not here for the previous session, I would just like to do a very quick introduction to Dr. Réka Howard. So Dr. Réka Howard is an associate professor of statistics and a director of consulting in the Department of Statistics at the University of Nebraska-Lincoln. She's also a courtesy professor in the Department of Agronomy and Horticulture at the University of Nebraska-Lincoln. Her research interests are in statistical consulting, statistical method development, and the application of statistical methods in plant breeding. As a director of consulting, she oversees the statistical cross-disciplinary collaboration and consulting lab at the University of Nebraska-Lincoln, which provides services including statistical analyses, interpretations, reports, visualization, helping clients to design the experiments. Her other research interest is in genomic prediction, focusing on the optimization techniques and machine learning implementations. She teaches courses in the areas of statistical consulting and statistical methods for MS and PhD students in statistics, agronomy, animal science, engineering, and related fields. Her talk today will cover practical introduction to factorial designs. And of course, we are very excited to have her here for the second session. So, Dr. Réka Howard, I will then hand over to you.
Thank you very much, Verena. Thank you, and thanks everybody for joining today. So yesterday we were discussing general concepts of designing an experiment. And today we will focus on factorial treatment design. So yesterday we talked about different treatment designs and experimental designs. And I mentioned that it's important to talk about both aspects when we talk about designing an experiment. So there are different types of factorial treatment designs. And I listed what we will talk about today, just double checking. Do you see my screen? So I'm moving pretty much, it's a Word file. So can you see a Word file? Hopefully.
Yes, we can.
Okay,
Yes.
perfect. So just briefly, I would mention several examples today. So the idea is that if we talk about these different types of factorial designs, then we can handle any combinations. What I mean by that, we will have qualitative and quantitative factors, and we will talk about how to deal with them differently. But first, let's talk about what's factorial arrangement of treatments. Why do we need this? So yesterday we briefly talked about this, that usually when we have an experiment and we want to answer some questions, we want to determine how a factor affects response. And many times we don't only want to focus on one factor, but several. So for example, yesterday I mentioned the irrigation and fertilizer example. So then we would have two factors. So it's important to distinguish between factors and factor levels. So in this case, when we are talking about irrigation and fertilizer, the fertilizer example, we are having two factors. And then if irrigation has, let's say, two levels or yes and no, then we are saying that it has two levels. And what's qualitative and quantitative? So we will start with qualitative factors. But before we start, I just want to mention why we are doing this. So as I mentioned yesterday, we could theoretically analyze these separately, but it wouldn't make any sense because then we couldn't look at the interaction. And that's the whole point, that we want to look whether the interaction is significant between factors or not. So I listed a whole bunch of descriptions of the factorial design and important facts, but I won't go through everything. So on purpose, I didn't create slides for this lecture. I am having this Word file because it has a lot of details that I won't talk about, but I'm happy to share this document. And if you are willing to go through and have questions, please let me know. I'm more than happy to help with the other details that I'm not covering today. So just very briefly, yesterday we saw this model description. Actually, we saw two different ways. First, we saw this way of describing a one-factor model. So where we had the response, we had an overall mean, and then we had the treatment effect and error included in the model. So this is called the treatment effects model. And somebody had this comment, and I was very happy to hear that when we talked about the residual, and it had a different formulation because it had a mu I. So instead of mu plus tau I, it just lumped together the mu and the tau I together. So these are fine, just different formulations. One's called cell means model, and the other one is treatment effects model. So again, these models, so describing the same thing, include one treatment. So what we do here is now we will start talking about two treatment factors, so A and B. So I will denote it by alpha and beta. You can use other letters. I just chose those. It doesn't matter what letters you choose. And then this is how you are including the interaction. So the alpha times beta is included too. So everything is explained in the document, what term describes what. So I mentioned yesterday that it's very important to talk about simple effects, mean effects, and interaction effects when we talk about factorial design. And I will talk about these in a little more details than yesterday to emphasize the importance. So we need to first look at what these three effects are. So what we will do is we will use this little example. I didn't even include data. I didn't include code. I just included some program outputs. So I will describe what's what, but let's just focus on the example. So let's say that we have a study and we want to look at the weight gain of rats. And let's say that we have four different diets, but how these four different diets are designed, that we have two different levels of protein, so high and low, and then two sources of protein, beef and cereal. So again, we are having a 2 by 2 treatment combination. So we have high and low protein level and beef and cereal. So that means that we have high-end beef, high-end cereal, low-end beef, and low-end cereal. That's why we are having the four levels. So we have two levels of each factors. And then let's say that we have 10 rats for each of the four treatment combinations. And I just included a little data output from, so I didn't include the original data set, but just let's imagine that this is the summary of our data. So the means of the combination, so the low beef, this is the low cereal, high beef, and then the high cereal. And then we can also look at the mean of these two, look at the mean of these two right here, and then the means of the beef and cereal at the low level here, and the beef and the cereal at the high level here. This is just an example. Don't worry about the numbers. They can be anything. So what we want to do is think about these simple effects, main effects, interaction effects. So what we do is how we calculate them and what the definition is. The simple effect is just the difference between two levels of a treatment factor at a given level of a remaining factor or factors. So in this case, we have remaining factor. So let's say that we want to look at a simple effect comparing beef and cereal. So I'm comparing beef and cereal at the low protein level, so at this level. So what I do is pretty much I am just looking at the difference of these two numbers, and that's it. So you have some more examples in the document how we get these. Do we have to do this by hand? Absolutely not. So your software, whatever you are using, will give you these numbers just so that you understand, and not just that you understand how you get these numbers, but which one to use. That's the most important, because any software will give you simple effects, main effects, but you have to know which one is the correct one to use. So this is when we are looking at the simple effects. So we are just comparing two factor levels at a specific level of the other factor. So we can look at the difference between the low and the high at the beef level too. And those examples are right here. And the main effects, don't worry about the formulas too much. Is just so the main effect is just the average of the simple effects. So let's go back to our little table. So we looked at the difference between beef and cereal at the low level, and we can look at the beef and the cereal difference at the high level. So we will get two numbers, and then the average of these two numbers will be the main effect. So that's what's happening here. So don't worry too much about the calculations, but more on the concept. But the details are there too. And the interaction, so it says here, the main effect is the average of the simple effects, and the interaction is the difference of the simple effects. So bottom line, how you can think about it is that when you are looking at the difference between the beef and cereal, in this case, the beef is lower than the cereal. In this case, when we are looking at the high protein, the beef is higher than the cereal. So that's why we start thinking, there is something going on, because depending on whether we have the low or high protein amount, the beef and the cereal difference will be different. So that's when we will start looking at or seeing interaction. But the interaction is by definition the difference in the simple effects. Again, you have these calculations. I'm skipping those, but they are there. So, but this is one important aspect that when you are actually plotting this interaction. So this is an interaction plot. So what's happening here is we have the high and the low protein, and then the different lines represent the different sources of protein, the beef and the cereal. So as we know, we have interaction when the lines are crossing. So this is nice, and we can see that there is an interaction. But one thing that I really want to point out, that just look at this plot. So what I did was I plotted the exact same data set. I did not change anything, but you saw that here I have high and low, and the two lines represent the sources. Now I flipped, and then here I have the sources, and the two lines represent the amounts, so the high and the low. And what I see is the lines are technically speaking not crossing. So when I really stretch this x-axis and then make the distance between the beef and cereal really, really long, then I can almost make this plot or this figure look like these lines are almost parallel. I'm not saying that they are parallel, but this is the issue when we are looking at one single plot, that this can be a little misleading, because when is the point when we say that there is an interaction? My point with this is when you have a data set and you see something like that, just plot it differently, and then maybe you will see something a little cleaner way to represent the interaction. Plus, you can also get the interaction effect from your output. Sorry that I'm moving back and forth because I thought that it was right there. So actually, in this case, when I run this in my software, I can see that the interaction is significant. So it's very clear that there is an interaction when I'm looking at this plot and I'm looking at the p-value associated with the interaction. But when I'm looking at this plot, and if I would only look at this plot, I would be a little hesitant, like, is it an interaction? Is it a significant, statistically significant interaction or not? So you have to be careful. That's my point with this example. This is just seeing how we could write this model. As you can see, now I don't have alpha and beta. I decided to say S for the source and A for the amount. So I have the source effect, the amount effect, and the interaction between the source and the amount for this example. And everything is described there. So this is my output for that example. So as you can see, the p-value, so this last column represents the p-values associated with the effects. So the interaction is the source by amount is 0.05. And this is actually very good because this is where we say, okay, if my magic number, so the alpha level is 0.05, is this significant or not? Because it's greater than 0.05, technically speaking. But be very careful. This 0.05 is just a chosen number. So if it's really close to 0.05 and it's greater, be careful because you'd rather say that there is a significant interaction than not, and then ignore and then just average over everything. You should not average over the simple effects. So what does it mean? So when there is an interaction, the difference between the beef and the cereal is different at the low level and the high level. That's what it means. So when you have a borderline significance, just treat it as a significant interaction. So this table just gives us the different combinations, the estimates, the mean estimates for the different combinations that we saw in the table. So don't worry too much about it. But then any of your softwares that you are using will give you the differences. So this is a low weird output, but this underscore means that I am looking at the beef and the high minus the beef low. So what I'm doing here, I am comparing the high and the low amounts at the beef source, and the estimate will be 20.8, and the p-value associated with it is significant. So this difference is significant. And this is a simple effect because I'm looking at the difference between high and low at a certain level of the source. But be careful. So what I said was it's not that you have to hand calculate these estimates and p-values because the software will give you these numbers. But when I am looking at, for example, I have to, yes, the third line. It says beef high, cereal low. Is it something that we are really interested in? No. So it's comparing the beef high with the cereal low. So what we do for simple effect, we are comparing two factor levels. So let's say the high and the low at a specific level of this other factor. So at, let's say, beef or cereal. But here I have beef and I have cereal. So this third line is, for example, something that I'm not interested in. So I have to be very careful knowing what I'm interested in, what I'm not interested in. And then this is just, again, another output of the interaction, and then this is my p-value associated with the interaction, and actually I am getting an estimate associated with the interaction. So this was an example where we had two levels of the each of the factors. So we had two factors, and they were qualitative factors. So qualitative meaning we had low and high, cereal and beef. So those were the two levels for each. So it was a 2 by 2 qualitative by qualitative factorial. So now we will look at another example, which will be similar. We are just increasing the factor levels. So this example will be qualitative by qualitative, but now we are increasing and we are saying it's a 3 by 5. So instead of a 2 by 2, we will have a 3 by 5. So what's the difference? So in this talk, I just want to emphasize the difference is how we analyze or we look at these examples differently and what we have to pay attention on. So in this example, actually, I got this example from a book called SAS for Linear Models. So we are investigating 3 seed growth promoting methods. So we will have 3 methods. So as I mentioned, it's a 3 by 5. So we have 3 methods. And we have 5 varieties. Okay, so again, these are qualitative factors. So 3 methods and 5 varieties. So we have 3 by 5, so 15 treatment combinations. So, and then let's say that we are having 90 yields measured. So I have the data set. I did not include a data set, but this example is based on a data set. So the model can be written like this. I forgot to mention last time, the assumption is the same as yesterday. So we have homogeneous variance, normally distributed error. So that's the same. And then every term is described. Here we have method effect, variety effect, and the interaction effect. So first, what you want to do, no matter what, plot your data, examine your data. So this is what's happening here. So we are having 5 varieties, and the different lines represent the different methods. So this blue is the A, and then we are having the red being the B, and then the C is this green one. So we can see that if we are looking at the response, which is yield, we want to have high yield. So the blue line is above the other two for every variety. So method A is significantly better than the other two. And then, so one thing that's interesting that we can notice some interaction going on potentially. So when we look at our output and we are first looking at the interaction, we can see that this is significant. But one thing that's different between the 2 by 2 case, that now this is just describing to me that there is an interaction somewhere. When I had a 2 by 2 case, I knew exactly where the interaction was because we have just 2 levels of the 2 factors. I can't have multiple interactions. But here, as you can see, we can have interaction between method A and variety 1 and 2. And I mean, we can have different interactions. So when you have multiple degrees of freedom associated with the interaction, in this case 8, because in our previous example it was 1, so for a 2 by 2 case it's 1, then we have to be careful with this p-value. One thing is, even if it's not significant, sometimes it happens that we have some interaction in our data. And then another thing is, even when it's significant, we don't know where it is. So we have to kind of dissect this interaction. So 8 degrees of freedom means that I can have 8 independent or orthogonal comparisons to investigate this interaction. So for example, I can just pick on this and say I want to compare the variety 4 and 5 for method B and C. I mean, there are different ways to come up with the 8 independent comparisons, but we have to be careful. That's the whole point here. I will be skipping some parts here just to make sure that we get to all of the examples. So I will point out that the output is comparing the different methods at certain varieties, and then you can take a look what's significant and what's not significant. So I am skipping these things because of time. So, but one thing too, since we are having 3 levels of the methods, so 3 methods and 5 varieties, we can have all kinds of comparisons. I did not talk about contrasts yesterday, and I'm not planning to do that today, but that's another important aspect, that sometimes you have to have specific comparisons that you are interested in. So in this case, let's say that I want to compare, for some reason, the average of methods B and C with method A for variety 1 and 2. So that's a very, very specific comparison. Then I have to actually hand code this. So the software won't just give you this specific comparison. So, but there are many, many comparisons that will give you, the software will give you without setting up contrasts. So I will kind of stop here with this example. So just one more thing that what we are doing here, sorry, let me just go back. No, here. So what we are doing in this output, we are looking at actually what I just mentioned. Uh, we are comparing method A with the average of B and C for variety 1, for variety 2, for variety 3, and for variety 4. So if you are going through these notes, one thing that you will realize that we don't have variety 5, and you are asking the question, why not? Because we had 5 varieties. Why isn't variety 5 included? So when you actually go here, to this output. So what we are doing here is we are comparing the method A and B, A and C, B and C for the different varieties. Okay. So what we wanted to do is look at the difference between uh, method A and the average of B and C. What did we say? We said that we can average two things when there is no interaction. Okay, so but at variety 5, so I said that variety 5 was not included. Why not? Because B and C, so B and C is actually, I'm looking at the wrong table, I'm sorry. B and C is significantly different for variety 5. So for variety 4, B and C is not significantly different. For variety 3, it's not significantly different, and so on. So variety 5 is the only one where B and C, so methods B and C are significantly different. If they are significantly different, I can't just average and say, oh, I want to average B and C and compare to A. So that's why in this output, variety 5 is not included when I am looking at the difference between method A and the average of B and C. So when you are actually putting these comparisons together, you really have to be careful what you can compare with and what you cannot compare. So far we talked about qualitative factorials, so a 2 by 2 and a 3 by 5. So now I will mention another other scenario when we are having a qualitative factor, but also a quantitative factor. So what does it mean? So this example will be a 2 by 3 qualitative by quantitative factorial. So let's just play with this example that's right here. So what we do is, sorry, I will talk about the example in a minute, but what we use when we have a quantitative, any quantitative factor, we will potentially we can use polynomials. So these are very good choice because one, they are very flexible, and usually we don't even have to include too many terms, and we like that because we are um, using some linear model. So what we do is, so they are simple and they are flexible. So, but one thing that you have to be very careful about when you are using polynomials, they are really not good at cyclic patterns. What does it mean? If you are having data set and it's a yearly data set, and you know that you are having a yearly pattern, it won't really work well because that's just now not what we use for not polynomials that you would use for a cyclic pattern. And another thing too, it's really bad for extrapolation. What does it mean? It means that if you have data, I'm just making this up, between day 0 and 100, you do not want to use this for predicting day 200 because it's not really a good choice. But in general, it works very well. So let's look at our example. So we had that we have a I have to go back, a 2 by 3 qualitative by quantitative example. So 2 levels for the qualitative factor and 3 levels for the quantitative. So what we do here, so this is the example right here. So the qualitative factor will be A, and that will have two levels, and the B will be the quantitative factor, and it will have three levels. So what we do here that a little different than before, one is we are doing regression now. So in your code, whatever you're using, software, you really have to be careful and uh, you have to specify that the B factor is not classification variable anymore. So the A is because that's a qualitative factor, but B is not. So in, for example, SAS, I have to specifically say that A is a classification variable in R2. And then I don't do that with B. So then my software will know that, oh, yes, B is a quantitative factor. Also, since we are doing regression, the default is a type 3 sum of squares. And then now I have to actually include a type 1 sum of squares for calculating our effects. So, and another difference is that now the interaction, how do I even handle the interaction? So when I have a quantitative factor, I have to look at how many levels I have. So I have three. It means that just think about the degrees of freedom. 3 minus 1, so 2. I can have up to two levels in my model. So I can have the linear and the quadratic effects. Okay, so two effects. And then, since I can have the linear and quadratic effect for the quantitative factor, I can have two interactions. I can have interaction between the linear effect of B, so the quantitative factor, with the qualitative factor A, and then I have the quadratic effect of B interacting with the qualitative factor A. Okay, so then this is how I write this out. So you could see that this was the regular polynomial way of writing the model. So I have intercept, I have the linear part and the quadratic part. So then I actually complicate this a little bit. I will include this I index, and then I have this BJ. So I'm actually including the qualitative factor A2. So again, we had three levels for B, so that's why we will have the intercept, and we will have the linear effect, and we will have the quadratic effect. And then on purpose, I actually left some code in my notes too for this example. So there is a specific example where we are evaluating some battery life. And what we do is we are having three temperature levels. So we are having 15, 70, and 125 Fahrenheit, which means that the temperature will be a quantitative factor. So this is my factor B that was in my previous example. And then we are having three plate materials. So we are having a qualitative factor with three levels and a quantitative factor with also three levels. So then what we do is, let's say that we want to see what effect do material and temperature have on battery life. So what effect do these factors have on the response? And is there any specific choice of material that would give uniformly long life regardless of temperature? Meaning, is there any interaction or not? And can I choose a material that's good no matter which temperature I am looking at? So since I have three levels of each factor, so first when I'm looking at the quantitative factor, I know that I have three levels, so I will look at the linear and quadratic effect. So for interaction, I will look at the interaction between material type, which is the qualitative factor, and the linear effect of temperature, and also the quadratic effect of the temperature. So same thing what we did before. And then this is just describing the model again. It's exactly the same as before, just it's explaining the actual terms with this battery example. So that can be useful. So this is the code. Don't worry, I didn't even mention SAS in my notes, not yesterday, not today, and that's not my point. But I just wanted to leave this in to show you what to do in general. Of course, your software can be very different, but what you have to pay attention on that in the class statement, so if you are specifying a classification variable, you would classify the material as classification, and you can see that I don't have to classify the temperature. That's how my software knows that that's not a qualitative factor, but that's a quantitative factor. And another thing is this type, H type equals 1, specifying that I will have type 1 sum of squares. So it depends on your software how you will actually tell the software, but probably you have to do that. And another thing is what terms do I enter into the model? So the material, the temperature is included. And then this is the most complicated part, like how do I decide what to add into the model? So as we said, the temperature was the quantitative part. So that's why I know that I have to add the interactions too. And so I'm adding the linear term of the temperature and the quadratic term of the temperature. And I am adding the linear interaction and the quadratic interaction. That's how I'm adding those. I know that I'm discussing a lot of things that where I am skipping details, and I feel bad about that. I just want to emphasize in this lecture what to pay attention, and I know that I'm not discussing details. So if you have questions afterwards, please feel free to contact me because I'm happy to help with these things. So this is just describing the output, and then what you do is, as you would do with a regression too, you have to decide which terms to keep and then rerun it with the final model. So in this case, this is a significant term, so the quadratic effect or the quadratic interaction. So if interaction is included, it means that when I have a different material, then the temperature will act differently on the response, which is the battery life. So it means that I cannot lump every material together. I cannot analyze a material A with material B with material C the same way. So I have to be very careful. So I have to separate them. So this is just saying what's my final model. Again, don't worry about code. Based on what's significant, I'm including those terms, and then I'm rerunning. And then I get estimates, and then I am, based on the estimates, I'm creating this plot. What's this plot? It's just the estimates for the battery life. So on the y-axis, we have the battery life. These are the different temperatures on the x-axis, and then these curves represent the different materials. So be careful, because we cannot say that this, which is material 3, is the best. This material 3 is the best above this temperature, which is 30-ish, let's call it 30. But below that point, actually, this material is better. So you have to be careful how you interpret the results. So the fourth example that I wanted to include is a quantitative by quantitative example. So now both of the factors are quantitative. So we will have a 3x4 example. So in this case, we are looking at metal and feed rates. So we have 3 feed rates and 4 depths. So both of the factor levels are, or both of the factors, sorry, are quantitative. So what we do is, now we are getting a little more complicated, or it seems like, and good news is that we don't have to, because based on the feed rate, so we have 3 levels, we could actually include the linear and quadratic effects because we have three levels. So then 3 minus 2 is 2, or 3 minus 1 is 2. So we would enter the linear and quadratic effects. But based on the four depths, so 1, 2, 3, 4, we would have three levels. So the linear, quadratic, and chordic. So we can add everything we want to, but it's kind of pointless to start like that. So what we do is we start with a simple model. Usually what we want to do is add the linear effect of every factor and the quadratic, because then we are capturing a linear effect and a curvature. So maybe we will need more. I'm not saying that this will be for sure our final model, but what we can do is just include linear effect of one, linear effect of the other factor, the quadratic effect of both, and then the linear interaction. And then you can ask, like, why not more? Because we want to be simple. And then what we do is this weird alpha, beta, ij represents a so-called lack of fit term. So we can add, and this is beautiful, we can add a term that captures everything else that we are not including that we should include. So we should actually include the chordic effect of the cuts. I did not include that. I didn't include a whole bunch of interactions that I should, because I'm hoping that this is not significant because this should capture everything else. So if it's not significant, I know that I don't have to go beyond this. But if this is significant, I have to actually add more terms. But again, when you plot your data, you see what's going on, whether you have curvature, what's going on, you can have more sense of what to include. But you don't want to overcomplicate your life. So you start simple and then see whether you need extra terms or not. So what you do is first you plot your data. Actually, when you plot your data, so again, this example is based on a data set, it looks like this. So what do I see? So let's imagine that this surface is a table, okay? And then this is a piece of paper. And so you can visualize this, that this is not parallel, and it has some curvature. So when I see some type of curvature, it means that, yes, the linear term won't be enough. So I need some type of curvature, and I'm hoping that the quadratic effect will be enough. And then if it's not parallel with this surface, it means that I might have some interaction going on too. So this is a good visualization because first I can see, like, okay, what type of model should I fit that's not overcomplicated. So what I do is I fit the model that I just mentioned, and then this lack of fit is, oh, not significant, so we are good. Then we don't have to put even more things into our model, so more terms. And then I can go again, oh, this term is significant, this term is significant, this is significant. So the d by d was not significant. So what I do is I rerun my model with the significant terms, and that's my final model. So these are just outputs, and then based on my model fit, this is what I get. And then this model actually captures what I was just saying based on the real data. I have curvature, and then I have this tilt, so it's not parallel to my table, so this surface. So I am pretty happy with this. So the fifth example that I wanted to talk about, and I'm sorry that I'm hurrying, but I just wanted to make sure that we capture every single combination of factorial design, so far we talked about two factors. So we had qualitative by qualitative, qualitative by quantitative, quantitative by quantitative, and then different levels. But now what do we do if we have more factors? Can we deal with that? Of course we can. So this example, so there will be an example or examples that are 2 by 2 by 2, qualitative by qualitative by qualitative. But now you can see that you can replace a qualitative with a quantitative, and you will be fine because you have all of these combinations and you know how to handle them. And you can have 4 factors, 5 factors. It doesn't matter after a point. So here, what we have to worry about that's different than what we had before is this three-way interaction. So far, we just had a two-way interaction. But again, if you have a four-way interaction, you will know how to handle this based on the three-way because you will see what we do. So what we do is we have three factors now. So let's say A, B, C. So far we just had A and B. So now we will have a B, C factorial, and then each, in this example, have two levels, but you could have more than two levels. So what we do is we include all of the main effects. We include all of the two-way interactions. So the A with B, A with C, B with C. And then we also include the three-way interaction. So what we do is, so first we look at the highest term in terms of the interaction, so the three-way interaction, whether it's significant or not. So I included three possible outputs. So the first output is when it's beautiful because the three-way interaction is not significant and none of the two-way interactions are significant. What does it mean? It means that I can average over the factor A, I can average over the factor B and the C. So I can look at the differences between the factor levels A, averaging over all of the levels of B's and C's. So I can use the mean effect. So when we have interactions, I cannot do that. So this is the first example where everything is beautiful. We don't have interactions. So then the second output represents a little more complication. So still, I don't have a three-way interaction. I don't have an interaction between B and C, A and C, but A and B. So now between A and B, I have an interaction. So what does it mean? It means that C is not included in an interaction. Okay, so C is not. So it means that I can compare C1 and C2. In this case, if we have two levels of C, we can see that since the degrees of freedom is 1, we have two levels of C. So it means I can actually look at the difference between C1 and C2 and average over B and A. So it's fine. But with A and B, I cannot do that. So what I can do is I can look at the difference between A and B, A1 and A1 and A2 at a specific level of B and C. That's what I can do. And then this is the third output, which is the most complicated one, where we are having a three-way interaction. So what do we do with a three-way interaction, and how do we interpret this, and how do we actually know that we have a three-way interaction? So when we have a three-way interaction, we have to be careful, and we have to use simple effects. So I cannot average over anything. I have to make sure that when I'm looking at the differences between A1 and A2, I look at them at specific level of B and C. So that's the interpretation part. But how do I actually visualize that three-way interaction is that I look at a two-way interaction. So I can pick A and B, okay, at C equals 1 and C1 and C2. So I just plotted two-way interaction plots. So I grabbed A and B, but I could grab B and C. I could grab A and C. It doesn't matter. So a two-way interaction plot at specific levels of the third factor. So in this case, everything has two levels. So A has two levels, B and C. So then what we do is I look at these two plots. And then how do I see, do I have an interaction here, a two-way interaction? Probably not. They look pretty parallel. Do I have an interaction here? Yeah, it looks like I have an interaction because they are crossing. So yes, for sure we have an interaction between A and B at C2. But we don't have an interaction between A and B at C1. So what does it mean? So I know that these represent two-way interactions, but how do I actually know whether I have a three-way interaction just looking at these? So what's interaction? Interaction really means that when I'm looking at differences at certain different levels, those differences will be different. So same things apply here too. If I'm looking at two-way interaction plots, if they are different, then I have a three-way interaction. So what does it mean? I don't have an interaction here. I have an interaction here. They act differently, totally different. then it means I have a three-way interaction. A tricky thing is, what if I would have an A-B interaction at C equals 1, so C1, and at C2? Actually, then I would have a two-way interaction between A and B at C1 and C2, fine, but that does not mean that I have a three-way interaction because they are acting the same no matter if I am having C1 or C2. So these two plots have to be different to say that we have a three-way interaction. Okay, so that's what I wanted to cover in a nutshell. I know that I missed a lot of details and I did not explain a lot of details that would have been important, but I really wanted to focus on the different ways of having factorials and what to pay attention on. So if you have questions now, please let me know. But if you have questions later, please email me. I know that several of you emailed me yesterday, and I mentioned that I will answer. So this week, I am probably not able to answer your questions. I just answered one person. But next week, I will make sure that I answer your questions. And then if you want to talk, we can set up time too. So please don't hesitate to contact me. But please forgive me that I will just answer you next week. So, any questions?
Awesome. Thanks so much for that very insightful lecture, Réka. I only see one question on the chat at the moment, but others, if you do have questions, please feel free to pop them on the chat or you can raise your hand and we can call on you when we are ready for you. So there's a question from Jeffline Kodzua that says, you mentioned that we can get in touch with you. Kindly share the email address again.
I just did.
Yeah,
Mhm.
I'll just copy it back onto the main meeting chat. There you go. Any other questions from the group? We'll give it a second or two in case there are some questions. Like I said, you're welcome to type in the chat or you can raise your hand. I see one hand raised. Mary, please go ahead.
Yeah, thank you so much for everything, Rita. Very nice. Thank you. I want her to go back to the page on simple effects for Factorial, just the two by two, the very first one.
Mhm.
Yeah. No, you have, yeah, the two by two. Yeah, just this one, beef and cereal. So I just want to, in clear terms, simple effect is what between maybe you say low and high, or maybe you say beef and cereal low. I want to be sure what I'm understanding. What is
Mhm.
simple, what is mean, and what is interaction?
Mhm. So, okay. So the interaction, let's start with the simple. The simple effect is just you are interested in, let's say, comparing the beef and the cereal, okay? But you have to actually say at what level of the other factors, in this case, factor, because we just have two factors. So I'm looking at the difference between beef and cereal at the low protein amount level. Okay, or I can also look at the difference between the beef and cereal at the high level. Or I can look at the difference between the low and high at the beef or low and high at the cereal. So I have actually four different simple effects. But when I'm comparing, let's say, beef and cereal, I can have two because low and high, at low and at high level. And when I'm comparing the low and high, also I can have two simple effects because one at beef level and one at cereal level. Okay. And then so when I'm talking about main effects, I can have two main effects because one will be comparing beef and cereal, but now I don't have to specify that at low or high. I just average, okay, the difference between beef and cereal and average of the low and high, okay? Or I'm looking at the difference between high and low, and I don't have to say add beef or add cereal, okay? Just think about your research and what you are interested in. So it's not beef, it's not cereal, not these things. But what you many times, and that's very hard, when many times we are just interested in, let's say, comparing the yield for using fertilizer and not using fertilizer, okay? And many times you say, I just want to know that. But if you have other factors in the experiment, and let's say irrigation, and you have an interaction between the irrigation and the fertilizer, you can't just say, oh, the difference between uh, um, having no fertilizer and fertilizer is blah on the yield. Because then the difference will be different whether you are using irrigation or no irrigation. Does it make sense? So that's why the simple effect and the main effect is important. So even if you don't remember these terms as simple effect, main effect, just remember, can I average the other factor levels or not? It depends on whether I have interaction. And the interaction will be just looking at the difference between the simple effects. So what does it mean? So again, don't worry about formulas, but just remember that interaction really means whether this difference between let's say, beef and cereal is acting the same way at the low level than at the high level or differently. Okay, because if they act differently, I can't say that, oh, beef is better, because beef is better where? At the low level or high level? Because on average, yes, we can say something, but it's kind of wrong, because at low, if it's lower, and then at high, it's higher, then I am missing some information. Does it make sense?
A bit. Thank you. A bit.
Oh,
Some
no problem.
sense.
Any other questions? And please, if you look at these notes and you have more questions, I know that I skipped details. Please feel free to email me, and I'm happy to meet with you guys on Zoom, and I'm happy to help. And then if you have questions, like you are analyzing your data and you're not sure, like I don't know what you were talking about, just please feel free to contact
I
me.
have another question.
Uh-huh.
When you say qualitative factors, are you referring to the character of the factor? Like, high is a character variable, and then the other one, low, is a character variable. When you come to R, I want to be sure what's qualitative and quantitative. Yeah.
So qualitative just means that you can classify. So it's not a number where, 10 is less than 20. So when you have classification variables, so let's say cereal and beef, low and high, even the low and high, I don't know what low is. Is it 10 or 20? High, 100 or 10? So there are classification variables. But if you can specify that, yes, your irrigation is 0, 10, 20, 50, 100, then those will be quantitative. Okay. And the difference is whether you can, so with the quantitative factor, you can specify whether you have a linear effect or a quadratic. So you can model that. With the qualitative, you can just compare these levels. That's the difference between the two.
And so qualitative doesn't have figures, doesn't have you don't say 10, 20, you just say high, low.
Yes.
But quantitative, you have it in quantified, like 10, 20
Yes, exactly.
degrees. Okay.
Mhm.
Okay. Thank you.
No problem.
Great. And then we've got one other question on the chat and just one quick comment, just giving you some praises, Réka. So I'll just read it out. It's from Paul Gibson. He's just saying, thank you, Réka, for an excellent presentation. The emphasis on polynomial regression is very worthwhile and often overlooked. Also, you exemplified the benefit of graphing things in multiple ways to understand the data in detail. So he's just singing your praises there a little bit. And then we've got one other question from Banele. Banele, you are welcome to unmute. I'll read your question and then you're welcome to follow up if you have additional clarification. So Banele says, I have a question. What will be my factorial when I have one cultivar, 5 inoculations, and 3 phosphorus levels.
So this is a 5 by 3. So the phosphorus levels, I'm assuming they are quantitative. And the inoculation, is it, so how do we specify the 5 inoculations? But it will
Oh,
be, sorry.
hello?
You can go ahead,
Yes, yes,
Vanille.
yes. Yes, yes. I just wanted to check when we're having that, as we're just trying to explain, if you have one cultivar, inoculations, it can be probably bacteria, probably you inoculate your seed with the bacteria, probably rhizobium. Then the fertilizer levels, it can be high, low, and intermediate. And also
Okay.
probably if we can re-explain in terms of the quantitative and the qualitative on that effect.
But do I understand correctly that both of the inoculation and the phosphorus levels will be qualitative? So you will say low, medium, high, and then just 5 different classifications for the inoculations.
Yes.
Yes, then it's a 5 by 3 because the one cultivar is just like, yeah, you have one. So it's a 5 by 3.
Oh, it's a 5 by 3. All right.
Mhm.
Oh, okay, okay, okay. So if it
Because you are
was
not
two, then if it was two cultivars, it will be two by
two
five
by.
by three.
Yes, exactly. Because now you're not comparing cultivars. You're just having one.
Oh, okay, okay, okay.
Mhm.
Thank you for that.
I want to say thank you to Paul. And I received an email from him. I'm sorry that I am saying it here, and I did not answer him yet because I was traveling, and I saw the email, and it's on my list to answer the email. And thank you, Paul. And I was very excited about your email.
No problem.
Great. Are there any other questions? I'm not seeing any other questions on the chat. There were a couple of questions about lecture materials, which we've shared just via the chat. We've uploaded them there. So anybody looking for the lecture slides for yesterday or this particular document that Dr. Réka has presented today, you'll find them in the chat. We can also, of course, email them around if you'd like. I do see two hands popping up now, so I'll go to Johannes first. Johannes, David?
Yes, thank you so much for the presentation. I quite learned a lot of things from the presentation. I just wanted to ask a quick question that if I'm testing 100 varieties of cowpea in a field that is naturally infested by parasitic weed, what type of experiment is advisable to use?
So you have 100 varieties, and then I'm sorry, what are you comparing other than the
Uh,
varieties?
I'm comparing the effects of a parasitic weed, which is Electra, on the varieties of cowpeas.
So, okay. So now we have to look at that. So, what's your response? And maybe I'm not getting what are the other factor or factors? Sorry, do you mind repeating what you just said?
Okay, so basically I have 100 varieties of
Mhm.
cowpeas, and I'm planting these varieties in a field that is naturally infested by parasitic weed, and I want to see the effect of that weed on the 100 varieties. So what type of design is
Okay, but are you controlling anything or you are just observing? Because
I'm just
that's, uh-huh.
observing basically. Yeah.
Because so one thing is when you are having treatments, like you need to like it has to be controlled, meaning you have to say, okay, I am giving this level of parasite to this variety and I'm giving that. So it's a little different because this is an observational study, not an experiment.
Yeah.
I'm not saying that you cannot analyze it, but you won't have an experiment here.
Okay. All right, thank you.
Oh, no problem.
All right.
Perfect. Then we've got one other hand raised from Francisca Mutunga. Please go ahead.
Hi, Réka. Thank you for the good presentation. I have a question from yesterday. When people were asking questions about how they can lay out their experiments or decide on what experimental design they would take, you had this question on how they were collecting the data, like is it once or over days? So I was wondering, why is that major consideration when choosing an experimental design? And two, I have an experiment with 3 varieties and 2 conditions, one with striga and one without striga. So that makes it a 3 by 2 factorial experiment, or yeah,
Yes.
thank you.
So, sorry, I will answer the second one because that's quicker. So yes, that's 3 by 2 because you have 3 varieties and 2 conditions. And then the first one, so why it's important, actually, we will talk about this next Tuesday. So it really matters whether it's not better or good, or it's good. So if you are repeatedly measuring the response on the same experimental unit, it's called repeated measures. And so far, the models that we talked about assumes that the measurements are independent, or the experimental units are independent. Okay, but now what we are doing is when you are repeatedly measuring the response on the same experimental unit, those measurements will be correlated. So the error structure will look different. So that's why to account for that is very valuable, because you want to really include that the measurements on the same unit are correlated. And it's very simple to actually do. You just have to include that. Does it make sense?
Yeah, it does. Thank you.
No problem.
Great. So I don't see any other hands raised. So last call for any final questions. Going once, going twice.
So,
Okay. Oh, go ahead, Panele.
sorry. So,
No problem.
just the last clarity. When I designed a CRD experiment for the glass house, for the question that I asked, because I'll do it on the pots. So how do I design that? Because I say that I'll be having 15 treatments.
What do you mean by I'm sorry, the 15 treatments?
Yes, so remember that I said I have one cultivar, then
Okay.
5 inoculation, and then three
Uh-huh.
phosphorus levels, yes.
Okay. So, okay, there are several things here.
Okay.
One is, I don't know if you have some pilot data. So do you know how many replications do you need?
Yes, 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 I need to do 3 replications.
So
Yes.
because replication will be important and randomization will be important. I don't know if blocking will be necessary. It's a good thing to block, and so I'm not against blocking, but it depends on whether it's necessary or not. So those are for this experiment, those are the things I would really consider. The randomization will be very important, and you will have replication, so that's good. Whether it's enough, I mean, it's hopefully. And then I would look if you need to or want to block. But this will be a pretty simple, straightforward experiment, I think. Not simple in a way that collecting data. I'm not making that a simple part, the stat part being simple, not what you are doing.
Okay. All right. Thank you.
Okay, perfect. Then Rachel, you've had your hand up. Do you want to go ahead?
Okay, yes. Thank you for your presentation. So I wanted to ask, what is the criteria for choosing to use a factorial design versus a split plot that you mentioned yesterday?
Yes, that's a very, very good question. So pretty much the resources. That's what it boils down to. Because for split plot, what you have is you have an experimental unit, and then you divide that experimental unit to evaluate the other experimental units. So, okay, this was a very bad way to say it. Sorry, I have to think about this. So with a factorial, you are looking at factor combinations. With a split plot too, but what you do is, do you remember that example that we had 3 different grasses in 1 year? And then the following year, within that field where we had the one grass, we divided that field and then we had the four or five, I don't remember, I think 4 varieties. So we don't have the whole chunk of field and say, okay, I had grass 1 with variety 1, and then another one with variety 2. So it's pretty much resource, like if you don't have the resources, and not just the resources, but sometimes it's convenience, like it's very hard to irrigate subsections of a field. It's much easier to just irrigate at a certain level for a bigger section. So that's how we decide whether we are using factorial versus split plot. Does it make sense?
Yes, it does. Thank you.
Great. I think we've got time for one more question if you're comfortable with that, Réka. We've got a couple of hands raised. So let's see how many we can manage before time is up. Natasha, you had your hand up next. You're welcome to go ahead.
Thank you very much. And thank you, Prof. Howard, for your nice presentation. I have a question related to repeated measurements on experimental units. So sometimes we might be interested, let's say we are working on crop plants, and let's say okra, for example. And this is a crop where you might be just interested at a specific growth stage, let's say flowering stage. So you are collecting your data just as at the flowering stage, let's say flowering and pod filling stage, because those 2 stages are kind of, they happen simultaneously when you are working on okra, for example. What I mean is, as soon as the flower comes up, the pod, once the flower drops today, you can have the pod tomorrow, and the pod is what you are interested in. It is the fruit. So when we are talking about repeated measurements on experimental unit, because from what I understood, you said that having repeated measurements on experimental unit could actually improve your data quality and analysis, if I got that well. So I want to ask, in such setup where I am just interested at a specific growth stage, which means I might not be having repeated measurements, but I have different traits, and it's just on a specific experimental unit. Do you actually think that that could amount to some good data analysis insights?
So do you mind if I ask back if I understand it correctly? So instead of repeated measures, you are having multiple traits being measured.
Yeah, like plant height, like the number of fruit. Yeah.
Yeah, that's totally normal. That's totally good. It's very good. Yes, it's not repeated measures because it's not over time, but you can take advantage of the multiple traits and look at the correlation between the multiple traits. But now the difference will be how you can analyze this differently, but you can, instead of ANOVA, you can do MANOVA. So what you can do is take advantage of the the multiple traits that you were measuring. And then you might want to consider them as response. So it's not that bad or good, it's just different. But yes, they can be correlated. But you don't analyze them with predictive measures.
Okay. I do have another question. It's about my question to you yesterday regarding my experimental design, where I'm having 320 okra sessions and 2 irrigation levels, just that the irrigation levels are in 2 separate trials. So I have the optimal condition where I water the plants normally, all the 320 sessions. And then I have the drought condition where I reduce the amount of water. So the difference is just the amount of water in both trials. So I would like to know, having 320 sessions and having these 2 irrigation levels, does that make it 2 by 320 factorial design? Even though the trials are in 2 separate, there are 2 separate trials?
Be careful, because so you're not replicating this, as I understand. Is that correct? Plus, you are assigning this irrigation level to the whole field, so that to the whole 320 sessions. Is that correct? So yeah, then there are several things that you have to be careful about. So one is that, if I understand it correctly, you are not replicating, but maybe you are, so please let us know. And another thing too, your treatment. So this is one thing that we talked about yesterday. That's important. What's your experimental unit versus sampling unit? So you are assigning that treatment, so the irrigation, to the whole field, and not to the individual plants or not individual varieties.
Yeah, to the whole field.
So that's why it's not. Yeah, sorry.
Okay, but in each trial, let's say for the optimal trial, I do have 2 replicates. So I have 320 times 2, so making it 640 plots for the optimal condition trial. And then I have the same for the drought condition trial. So I just wanted to find out if that is still, yeah.
So just so that we understand it correctly. So you have the optimal trial in a totally separate field, and then you have the drought trial in a totally separate field, and you have the 2 replications within the fields. Is that correct?
Yeah, they are just, the trials are next to each other, separated by a 4-meter buffer, just to avoid water interference.
Okay, so they are together. Pretty much. So one field, so do I understand? So you have one field, you have rep 1, rep 2, rep 3, rep 4. Rep 1, 2 are getting irrigation, and rep 3, rep 4 are not getting irrigation.
Exactly.
Okay. So the only thing that you have to be careful is that they are together. So, I mean, yes, you can compare, but you have to be careful whether there is a field effect or not. So, and you want to account for that too, that the rep 1, rep 2 not getting irrigation are together, and then do you understand what I mean? That let's say that one, I don't know how the layout is, but let's say that one part of the field has better soil or something is going on, since there is no randomization, but yes, still you can compare the irrigation with the no irrigation.
Okay, but I did have randomization because I use R to randomize all 320. So yeah.
So those, yeah. So yes, but just I would look at the field conditions too if there is anything, but yes, I would not worry about that.
And it was under a drip irrigation system. So for the drought stress, we just stop the water to run into that block. And then for the optimal condition, we water throughout the season. So
Mhm.
yeah, it was something like that. So is that still a 2 by 320 factorial design?
I would have problem when we are analyzing the 320. That like whether you are getting estimates because you are having two reps and mm, that's when it comes into picture of potentially grouping the like I would allow more than just run a 320 by 2 factorial. So I would potentially look at those genotypes and try to cluster them too and look at what your results are. So do a lot of exploratory data analysis before just fitting a model.
Okay. Okay. Thank you very much.
Oh, no problem. I saw that Paul mentioned, yes. So I wish we would talk about split plot too. So the split plot on Nova looks different than Factorial. So you have an extra error structure. So you are having, instead of just the residual term, sorry, I'm just talking about the split plot question, going back to the split plot. So how do you decide between split plot and Factorial? So you have, so there are several things that are different between Factorial and split plot. So you have two, if it's true split plot, you have two different experimental units and you have two levels. So it's a hierarchical structure. It has a hierarchical structure. So the ANOVA will look different because you will have a so-called main plot error and a subplot error. The subplot error is the same as the normal residual for a factorial, for example. But I mean, that's our regular residual. But the main plot error has to be also included in an ANOVA. So it looks different and analyzed differently. But thank you for the note.
Great. So I do want to say that we are just about out of time at the moment. We had one other hand raised. Mary, if you have a quick question, you can go ahead. Sure.
Thank
Go
you.
ahead.
Just a clarification, because you have the 5 by 4 as a factorial, two-way factorial. And then you are also moving to polynomials. At what stage do you move it? You just analyze prompt normally and you have your y is equal to the overall mean plus all the effects of the two factorial. But I'm seeing that you're moving the same two-way ANOVA and you're having polynomial regression. How do you determine that?
So the polynomial comes into the picture when you have quantitative, any quantitative factor. That's when the polynomial comes in. When you have only qualitative, you don't have polynomial. But the polynomial comes into the picture because you are having these not classification, but quantitative variables, so the numbers. And that's when you can actually model the linear, quadratic, cubic, whatever relationships. So that's even if you have one or two factors that are quantitative, you will start working with the polynomials.
Okay, thank you.
No problem.
Great. So I think that then brings us to the end of the session. We are starting to lose a few folks. A few people are dropping off. So yeah, just to say again, very, very much thank you to Dr. Réka Howard for delivering these two lectures. She's got another lecture next week, so one more to go, and then we'll wrap up her module. She has shared her email address, so any burning questions or any other questions you have about project or experimental design, you're all welcome to reach out to her as noted. Thanks, Sumir. Sumir's just popped lecture 3's information onto the chat. It will be on the 16th of June, so next week, Tuesday at the same time. And we'll talk about block and repeat measures. So thanks, everybody. We'll see you next week, Tuesday. Thank you, Réka.
Thanks, everyone.